Genome editing applications. As of 2012, efficient genome editing had been developed for a wide range of experimental systems ranging from plants to animals, often beyond clinical interest, and was becoming a standard experimental strategy in research labs. The recent generation of rat, zebrafish, maize, and tobacco ZFN-mediated mutants and the improvements in TALEN-based approaches testify to the significance of the methods, and the list is expanding rapidly. Genome editing with engineered nucleases will likely contribute to many fields of life sciences, from studying gene functions in plants and animals to gene therapy in humans. For instance, the field of synthetic biology, which aims to engineer cells and organisms to perform novel functions, is likely to benefit from the ability of engineered nuclease to add or remove genomic elements and therefore create complex systems. In addition, gene functions can be studied using stem cells with engineered nucleases. Listed below are some specific tasks this method can carry out. Targeted gene mutation. Gene therapy. Creating chromosome rearrangement. Study gene function with stem cells. Transgenic animals. Endogenous gene labeling. Targeted transgene addition. Targeted gene modification in animals. The combination of recent discoveries in genetic engineering, particularly gene editing, and the latest improvement in bovine reproduction technologies, e.g. in vitro embryo culture, allows for genome editing directly in fertilized oocytes using synthetic highly specific endonucleases. RNA-guided endonucleases, clustered regularly interspaced short palindromic repeats associated Cas9, CRISPR/Cas9, are a new tool, further increasing the range of methods available. In particular, CRISPR/Cas9 engineered endonucleases allows the use of multiple guide RNAs for simultaneous knockouts, KO, in one step by cytoplasmic direct injection, CDI, on mammalian zygotes. Furthermore, gene editing can be applied to certain types of fish in aquaculture such as Atlantic salmon. Gene editing in fish is currently experimental, but the possibilities include growth, disease resistance, sterility, controlled reproduction, and color. Selecting for these traits can allow for a more sustainable environment and better welfare for the fish. AquAdvantage salmon is a genetically modified Atlantic salmon developed by AquaBounty Technologies. The growth hormone regulating gene in the Atlantic salmon is replaced with the growth hormone regulating gene from the Pacific Chinook salmon and a promoter sequence from the ocean pout. Thanks to the parallel development of single-cell transcriptomics, genome editing, and new stem cell models, we are now entering a scientifically exciting period where functional genetics is no longer restricted to animal models but can be performed directly in human samples. Single-cell gene expression analysis has resolved a transcriptional roadmap of human development from which key candidate genes are being identified for functional studies. Using global transcriptomics data to guide experimentation, the CRISPR-based genome editing tool has made it feasible to disrupt or remove key genes in order to elucidate function in a human setting. Targeted gene modification in plants. Genome editing using meganuclease, ZFNs, and TALEN provides a new strategy for genetic manipulation in plants and are likely to assist in the engineering of desired plant traits by modifying endogenous genes. For instance, site-specific gene addition in major crop species can be used for trait stacking, whereby several desired traits are physically linked to ensure their co-segregation during the breeding processes. Progress in such cases have been recently reported in Arabidopsis thaliana and Z. maize. In Arabidopsis thaliana, using ZFN-assisted gene targeting, two herbicide-resistant genes, tobacco acetolactate synthase SURA and SURB, were introduced to SUR loci with as high as 2% transformed cells with mutations. In Z. maize, disruption of the target locus was achieved by ZFN-induced DSBs and the resulting NHEJ. ZFN was also used to drive herbicide tolerance gene expression cassette, PAT, into the targeted endogenous locus IPK1 in this case. Such genome modification observed in the regenerated plants has been shown to be inheritable and was transmitted to the next generation. A potentially successful example of the application of genome editing techniques in crop improvement can be found in banana, where scientists used CRISPR/Cas9 editing to inactivate the endogenous banana streak virus in the B genome of banana, Musa species, to overcome a major challenge in banana breeding. In addition, TALEN-based genome engineering has been extensively tested and optimized for use in plants. TALEN fusions have also been used by a U.S. food ingredient company, Calyxt, to improve the quality of soybean oil products and to increase the storage potential of potatoes. Several optimizations need to be made in order to improve editing plant genomes using ZFN-mediated targeting. There is a need for reliable design and subsequent test of the nucleases, the absence of toxicity of the nucleases, the appropriate choice of the plant tissue for targeting, the routes of induction of enzyme activity, the lack of off-target mutagenesis, and a reliable detection of mutated cases. A common delivery method for CRISPR/Cas9 in plants is agrobacterium-based transformation. tDNA is introduced directly into the plant genome by a T4SS mechanism. Cas9 and gRNA-based expression cassettes are turned into T plasmids, which are transformed in agrobacterium for plant application. To improve Cas9 delivery in live plants, viruses are being used more effective transgene delivery. Gene therapy. The ideal gene therapy practice is that which replaces the defective gene with a normal allele at its natural location. This is advantageous over a virally delivered gene as there is no need to include the full coding sequences and regulatory sequences when only a small proportions of the gene needs to be altered, as is often the case. The expression of the partially replaced genes is also more consistent with normal cell biology than full genes that are carried by viral vectors. The first clinical use of TALEN-based genome editing was in the treatment of CD19 plus acute lymphoblastic leukemia in an 11-month-old child in 2015. Modified donor T cells were engineered to attack the leukemia cells, to be resistant to alemtuzumab, and to evade detection by the host immune system after introduction. Extensive research has been done in cells and animals using CRISPR-Cas9 to attempt to correct genetic mutations which cause genetic diseases such as Down syndrome, spina bifida, anencephaly, and Turner and Klinefelter syndromes. In February 2019, medical scientists working with Sangamo Therapeutics, headquartered in Richmond, California, announced the first-ever in-body human gene editing therapy to permanently alter DNA in a patient with Hunter syndrome. Clinical trials by Sangamo involving gene editing using zinc finger nuclease, ZFN, are ongoing. Eradicating diseases. Researchers have used CRISPR-Cas9 gene drives to modify genes associated with sterility in A. gambiae, the vector for malaria. This technique has further implications in eradicating other vector-borne diseases such as yellow fever, dengue, and Zika. The CRISPR-Cas9 system can be programmed to modulate the population of any bacterial species by targeting clinical genotypes or epidemiological isolates. It can selectively enable the beneficial bacterial species over the harmful ones by eliminating pathogen, which gives it an advantage over broad-spectrum antibiotics. Antiviral applications for therapies targeting human viruses such as HIV, herpes, and hepatitis B virus are under research. CRISPR can be used to target the virus or the host to disrupt genes encoding the virus cell surface receptor proteins. In November 2018, Jiankui announced that he had edited 2 human embryos to attempt to disable the gene for CCR5, which codes for a receptor that HIV uses to enter cells. He said that twin girls, Lulu and Nana, had been born a few weeks earlier. He said that the girls still carried functional copies of CCR5 along with disabled CCR5, mosaicism, and were still vulnerable to HIV. The work was widely condemned as unethical, dangerous, and premature. In January 2019, scientists in China reported the creation of 5 identical cloned gene-edited monkeys, using the same cloning technique that was used with Zhong and Hua, the first-ever cloned monkeys, and Dolly the sheep, and the same gene-editing CRISPR-Cas9 technique allegedly used by Jiankui in creating the first-ever gene-modified human babies Lulu and Nana. The monkey clones were made in order to study several medical diseases. In the near future, the new CRISPR system will also be able to eradicate diseases and conditions that humans are predisposed for. With this new technology, scientists will be able to take the genes of a human sperm cell and egg, and replace the genes that activate cancer or other abnormal or unwanted defects. This will take the stress off from parents worrying about having a child and not able to live a normal life. After just one generation of this process, the entire future of the human race would never have to worry about the problems of deformities or predisposed conditions. In the future, an important goal of research into genome editing with engineered nucleases must be the improvement of the safety and specificity of the nuclease's action. For example, improving the ability to detect off-target events can improve our ability to learn about ways of preventing them. In addition, zinc fingers used in ZFNs are seldom completely specific, and some may cause a toxic reaction. However, the toxicity has been reported to be reduced by modifications done on the cleavage domain of the ZFN. In addition, research by Dana Carroll into modifying the genome with engineered nucleases has shown the need for better understanding of the basic recombination and repair machinery of DNA. In the future, a possible method to identify secondary targets would be to capture broken ends from cells expressing the ZFNs and to sequence the flanking DNA using high-throughput sequencing. Because of the ease of use and cost efficiency of CRISPR, extensive research is currently being done on it. There are now more publications on CRISPR than ZFN and TALEN despite how recent the discovery of CRISPR is. Both CRISPR and TALEN are favored to be the choices to be implemented in large-scale productions due to their precision and efficiency. Genome editing occurs also as a natural process without artificial genetic engineering. The agents that are competent to edit genetic codes are viruses or subviral RNA agents. Although GEEN has higher efficiency than many other methods in reverse genetics, it is still not highly efficient. In many cases, less than half of the treated populations obtain the desired changes. For example, when one is planning to use the cell's NHEJ to create a mutation, the cell's HDR systems will also be at work correcting the DSB with lower mutational rates. Traditionally, mice have been the most common choice for researchers as a host of a disease model. CRISPR can help bridge the gap between this model and human clinical trials by creating transgenic disease models in larger animals such as pigs, dogs, and non-human primates. Using the CRISPR-Cas9 system, the programmed Cas9 protein and the sgRNA can be directly introduced into fertilized zygotes to achieve the desired gene modifications when creating transgenic models in rodents. This allows bypassing of the usual cell targeting stage in generating transgenic lines, and as a result, it reduces generation time by 90%. One potential that CRISPR brings with its effectiveness is the application of xenotransplantation. In previous research trials, CRISPR demonstrated the ability to target and eliminate endogenous retroviruses, which reduces the risk of transmitting diseases and reduces immune barriers. Eliminating these problems improves donor organ function, which brings this application closer to a reality. In plants, genome editing is seen as a viable solution to the conservation of biodiversity. Gene drive are a potential tool to alter the reproductive rate of invasive species, although there are significant associated risks. Human enhancement. Many transhumanists see genome editing as a potential tool for human enhancement. Australian biologist and professor of genetics David Andrew Sinclair notes that the new technologies with genome editing will allow it to be used on individuals to have healthier children, designer babies. According to a September 2016 report by the Nuffield Council on Bioethics, in the future it may be possible to enhance people with genes from other organisms or wholly synthetic genes to, for example, improve night vision and sense of smell. George Church has compiled a list of potential genetic modifications for possibly advantageous traits such as less need for sleep, cognition-related changes that protect against Alzheimer's disease, disease resistances, and enhanced learning abilities, along with some of the associated studies and potential negative effects. The American National Academy of Sciences and National Academy of Medicine issued a report in February 2017 giving qualified support to human genome editing. They recommended that clinical trials for genome editing might one day be permitted once answers have been found to safety and efficiency problems, but only for serious conditions under stringent oversight.